Okay, so let's look at the first part of question 5, right? So let's look at the question a bit more carefully. It says to explain how modifying the proportion of cis to trans fatty acids, blah, 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 right? So many of you, right, and that's really many of you have misread the question, right? Modifying the proportion of fatty acids is not the same as modifying cis fatty acids to trans fatty acids, right? Modifying the proportion can go both ways, right? You can increase the proportion of the cis fatty acid, which means to modify or convert or change the trans fatty acids to cis. So you have more cis, right? Or it can be to increase the proportion of the trans, which is the opposite, right? So modifying the proportion actually can go both ways in terms of the changing of the fatty acids. It can be trans to cis or cis to trans. But for many of you who have misread the question, you have restricted yourself to just one kind of conversion, right, or change, right? You thought that the question is talking about changing the cis fatty acids to trans, which is wrong, actually, right? So when you have misread the question from the get-go, you are already on the wrong path. Yeah? So read the question carefully, yeah? And a bit more, right, reading the question is talking about fatty acids in the cell membrane, right? That means fatty acids in the phospholipids, right? Each phospholipids molecule has got 2 fatty acids, right? So these are the fatty acids that the question is asking for, not fatty acids in the triglycerides, in the fat tissue, right? There's no fat tissue in bacteria anyway, right? It's the fatty acids in the membrane that the question is asking about, yeah? And the context of the question is when the temperature drops. That means membrane fluidity will decrease because of the drop in temperature. So the modifying of the proportion of the fatty acids, therefore, is to increase membrane fluidity so that the membrane fluidity doesn't decrease so much, right? You should combat or counter, right, the decrease due to the drop in temperature so that the membrane retains a level of fluidity, which is necessary, right, for membrane's function. Okay? So then, right, once you have deduced this by reading the question, you want to look for what is the modification in the proportion of fatty acids that will increase the fluidity of the membrane, right? Then you look at the information given, which fatty acid will increase the membrane fluidity. That is obviously the cis fatty acid with the cis double bond that creates a kink or a band in the fatty acid tail, yeah, which is something that you have learned before, right? What we have learned before is between saturated and unsaturated fatty acids, right? One have no double bond, so it's linear. The other one has a double bond, so there's a kink. That's what you learned before. Now the question is showing you something different. Both fatty acids, cis and trans, are unsaturated, but yet the trans fatty acid is not giving you a kink. It's the cis fatty acid with the cis double bond that is creating a kink. Nonetheless, right, the application of what you have learned here is you need to have a kink to increase the membrane fluidity, right, which is what you have learned before, right? When you have a kink in the fatty acid tails of the phospholipids are further apart, therefore the membrane is more fluid. Okay, so this is the application of both the information in the figure for the two fatty acids with the two types of double bonds. Then you link it to the content knowledge that you have learned before in terms of the kink and the increase in fluidity because of the kink. Okay, so that's why the answer is like that. I'll just leave you to read the answer yourself. Okay, I'm not gonna repeat the answer. I've already explained most of the stuff already. Okay, so next up, I'm just gonna show some very common and really alarming kind of mistakes, right? So firstly, in terms of the exam skills, in terms of reading the question, right, understanding the context of information, okay, it's mentioned already from the start, right, that many of you are misreading the question, okay? And here, some of you maybe don't know what a kink is and somehow misread the information and concluded that the trans fatty acid has a kink and not the cis. So a kink is a band in the fatty acid tail, which is clearly in the cis, not the trans. Okay, then more alarmingly, right, there are so many of you who have the confusion or the lack of familiarity with the content, right? These are some of the very common, very alarming mistakes, right? Some of you mentioned that when the temperature is low, it is better for the fatty acids, for the phospholipids to pack closer together, which is BS, right? Total opposite of what you have learned. Because when temperature is low, the fluidity of the membrane needs to be increased to counter the decrease in the fluidity due to the low temperatures. Yeah? If not, your membrane will freeze solid and you crack, right? Then somehow, some of you mentioned, right, the trans fatty acids has no kink, right, which is correct. But then somehow you can say that that will increase membrane fluidity. That is again total BS. Total opposite of what you have learned. It's the kinks that increase membrane fluidity, right, not the absence of kinks. Okay, then this one also quite strange that some of you mentioned the membrane when it is packed closer together, when it's less fluid, somehow it can trap heat better. Since when the membrane traps heat, phospholipids don't function to trap heat, right? Trapping heat is the function of triglycerides, which is found in the fat tissue under our skin or around our organs. Bacteria don't have skin or organs. Membrane doesn't trap heat. So you mixed up, right, all the content under lipids. And this one also very strange that so many of you have written that there is a decrease in surface area when there are kinks. The length of the fatty acid tails are the same with or without the kinks. So why would there be a difference in the surface area? It's the same, right? The kinks increase fluidity because the kinks physically push the phospholipids further apart in the picture that I've shown earlier, right? The phospholipids molecules are just further apart because of the kinks. They are further apart, not that there is decreased surface area for bonds to form. And this decreased surface area has not appeared anywhere in the notes or whatever. I don't know where you get it from, but it is not correct. Okay? So when so many of you, and really there are so many of you that have made all these mistakes, especially, right, more alarmingly in the contents for in a topic that is covered in J1, and you have time to actually revise, then that is very alarming to me, and that's why I sent the email earlier asking you that maybe you should retry the CA1, right, in an open book manner, so that you can better deduce for yourself whether it is the contents that you have a problem with. Because if it is the contents that you have problem with, then there's no need to talk about exam skills, application, or whatever, right? You don't even have the contents. There's nothing else that we can talk about. You need to revise the contents first. For those of you who have made all these very, very, very basic and really like total opposite of what you should have learned kind of mistakes, then that is very alarming. Yeah? Okay, so anyway, yeah, that's 5A part 1, yeah?